some of you know me already because I'm really a fan of the school. And always try to participate at the science education of the kids. And I think this is part of it, of course, as well. So today, the presentation is entitled Alien versus Predator. You can see already these two creatures on the screen because it really very well describes what happens during a human infection by the parasite Leishmania. Most of you may never have heard about Leishmania, so the only aim of my talk is that when we're done, you can tell someone, oh, I know what Leishmania is. So that's the only aim. You don't have to understand more than that. To spin it a little bit further, so Alien versus Predator. Hang on, I think, yeah. So this is like a movie plot. And of course, the actors of this movie plot is, first of all, the alien. And in this case, it is this parasite Leishmania that is just one single cell shown here in the lower part of the slide. I think you may see my mouse. This is this oval-shaped parasite that has a long protrusion, which is a flagellum that allows this parasite to be mobile. And so why is this the alien? Well, first of all, if you look at the parasite itself, its biology, these parasites are eukaryotic organisms. I guess that most of you know the difference between a prokaryote and a eukaryote. Prokaryote DNA is floating in the cytoplasm, and in a eukaryote, you have a nuclear membrane that encapsulates the DNA. And so this is a eukaryote, just like we are eukaryotic organisms as well. And so usually all eukaryotes, they have a lot of biology in common, and this makes it difficult to combat these parasites because they're eukaryotic cells just like we are. And whatever kills these cells most of the time is very toxic to us as well. However, over the years when we study these parasites, we have come to the conclusion that this must be an extraterrestrial, because there's so many differences. You know, we don't really know why and how these parasites evolved so much different biology. Anyways, a first funny little hint about being an alien is, of course, that the parasite has its own spaceship. And this is the little phlebotom sandfly that can be infected with this form of the parasite. And during a blood meal, these parasites are then injected into any kind of mammal, including humans, of course. So the parasite is not human specific, but can infect actually any kind of vertebrates. The more, I would say, extraterrestrial biology is shown here on this picture. I won't go into details. I will just tell you a few things that are quite unique to these parasites. So first of all, this is an electron microscopy image where the parasite is cut through, and then you can see all this grainy stuff, which is the cytoplasm and the nucleus in the middle. So the unique features of these parasites are, for example, a surface coat. We're going to talk about the surface coat in just a few minutes, that covers the entire parasite, and it's really like a shield against any kind of attack from the immune system, anything that is a chemical attack. These parasites are shielded. Of course, because this covers the entire parasite, normally organisms interact with their environment to eat and, of course, to release some metabolic end products. And so the parasite has invented this structure here where the flagellum comes out, so this motility organelle, which is called the flagellar pocket, which is the exclusive site of any kind of exchange between the parasite and the environment. Now, this flagellum is anchored in this structure here. I don't know if you can see very well. It looks like a little smile. This is a very compacted DNA that doesn't belong to, well, it's not the parasite DNA, it is the mitochondrial DNA. Now, mitochondria, you remember from class, are the energy house of the cell, and Leishmania has one gigantic mitochondrion which goes throughout the entire cell, and this is the DNA, and usually this DNA, at least in our cells, is just a ring. And in Leishmania, and we don't know exactly why, there are thousands of rings, and I show you here this little insert here, thousands of rings that are concatenated, which means really they are interlinked like in a chain. And so we don't understand why the parasite has these structures, what they're doing, and when the parasite divides, how can you divide such a complicated structure? We have no idea. The next little insert are glycosomes, which are absolutely specific organelles for glycolysis in these parasites. And another very, very unique feature of these parasites, which truly always baffles my colleague scientists, I'm not so sure about you, but these parasites, even though they have a nucleus and they have the DNA and they're eukaryotes, they do not express their DNA like we do. They just express the DNA all the time. There is no regulatory elements in the DNA that turns on and turns off genes. All the genes are expressed all the time, which is quite unique because normally you don't want to express what you don't need. That's why we have developed promoters and transcription factors. If anyone has a question, you know, you can interrupt me, it's not a problem. So the second player of the alien versus predator plot is, of course, the predator, which in my story is represented by the macrophage, which is a white blood cell that is known to be very active in eating any kind of foreign material that is in our tissues or in our blood. Now, I told you that Leishmania, of course, is introduced into our blood system through the bite of these insects which are infected. And so the first cell they encounter are these macrophages. And here's just an EM image that I picked from the internet because it looks a little bit like the predator. So here is an electron microscopy image where you can see a macrophage with a very ruffled kind of surface that is on the way to engulf this Leishmania parasite. And usually macrophages are known to be the most toxic cells in our body. They are designed to eat and to destroy and digest. And so at this moment, I always tell people like these parasites, they even attract macrophages as if they were suicidal. But of course, they attract these macrophages because these parasites have evolved to perfectly well adapt and survive in these very toxic cells. And so this is just a schematic view. You see a little black parasite coming in here. And so these parasites, they really have to survive many different levels of attack inside the macrophage. So already when they're taken up here, this EM, already when they're taken up, there are toxic oxidants that are produced at the site of uptake, which normally can kill many bacteria and other organisms, but not Leishmania, I told you, has this very protective sugar coat or lipid coat that protects the parasite. Then once the parasite is inside, it is in this kind of vesicle, or we call this a vacuole. And this vacuole starts to fuse with these digestive vesicles called lysosomes. And so that's why it turns a little bit red. And this is literally what we have in our stomach, which means acidic enzymes that just degrade everything. And again, the parasite, thanks to his shield, is not affected by this very toxic environment. But on the contrary, the parasite starts to manipulate the host cell. And this is why Leishmania is such a bad disease, because it sits inside a very essential immune cell, the macrophage, which normally is at the basis of any kind of immune response. And here, the parasite deactivates, literally, paralyzes these macrophages, and even hides inside these immune cells. And so this is why we have such a big problem with Leishmania. And so inside these macrophages, these long-shaped cells with the flagellum, they turn into a different developmental stage that is characterized by a more rounded shape and a tiny little stumpy flagellum, which most likely anchors the parasite inside this vacuole. Then, of course, in every kind of action movie, unfortunately, there is a lot of collateral damage. And in our case, the collateral damage, you guess it, is us, because these two, predator and alien, fight in our body. And so here's just a few pictures of the typical symptoms or pathologies that are caused by Leishmania parasites. You have a cutaneous form where people that are infected in the skin develop these huge lesions, which are normally self-healing, but it takes months and months. On the right side, you have a poor person that is infected with a parasite that goes to the mucosal tissue in the nose. And again, there is a lot of tissue damage in the nose. And in the middle, you have a visceral involvement where the parasite goes towards the liver and the spleen. And this poor boy, what you see here taped is the spleen, which is like 10 times the size of a normal person. So all of these diseases are not caused directly by the parasite. They are caused by the effect of the parasite on your immune system. It's what we call immunopathology. So your immune system goes crazy and attacks even your own tissue. So, of course, you know, producing a nice movie, we need a film studio. And in our case, this film studio is shown here. It's the Institut Pasteur, where I'm sitting here right now in the middle of the 15th arrondissement. So we have, of course, the original old building. We have super fancy modern buildings. It's a huge campus. 3,500 people are working here. I don't know how many buildings we have, but we have, just to tell you, 12 departments, 130 labs, and 10 Nobel Prizes so far. I'm not going to go through all of these things. It's a little bit boring, but the best part of the Pasteur Institute is shown here in the lower map, which is actually, we are the mothership of 33 Pasteur Institutes worldwide. And I have the pleasure to work with many, many of these institutes, which of course comes with travel and visits and, you know, food and things like that. So it's really very enjoyable. So, of course, we need a production team to make this plot, this movie happen. And so this is my team right now. So we have about 12 people. The team is divided in 3 sub-teams that investigate different aspects of Leishmania macrophage interaction. And finally, of course, you have a producer, and I have to say it's me. You know? I think Anna told me to just mention briefly how I arrived in Paris at the Pasteur Institute. And so this is my little life cycle here. I studied at the university in Mainz in Germany. You recognize my German accent, of course. And obtained a master in immunology. Then I went for a PhD in development in Pasteur already in Paris. Then I went to St. Louis, Missouri, where I switched into parasitology, and I stayed there. I got my first laboratory at the New York University in the US. And then, of course, in 2005, I came back as head of a lab to the Pasteur Institute in Paris. So this is my now concluding, I would say, the more funny introduction. I just want to give you a little bit more feedback on Leishmania. As you've seen before, we distinguish different pathologies, and these different pathologies are, first of all, geographically kind of localized, and they're also linked to different types of Leishmania parasites. So you have, for example, here in the Middle East, cutaneous Leishmaniasis, which gives you the skin lesion, which is caused by these 2 different types of Leishmania, major and tropica. You have the very bad, even deadly disease, the visceral Leishmaniasis, which is endemic in India and in East Africa. And then you have this mucocutaneous Leishmaniasis, which even on old mummies, we can find the traces of the tissue destruction here in the upper jaw, which is more endemic in South America. We have, of course, many animals that get very sick, and unfortunately, dogs worldwide are extremely susceptible to Leishmania, and they are what we call a reservoir of the disease. You could vaccinate and treat every human. You will never eliminate Leishmaniasis because there's so much parasite in animals, and you can just not treat the animals. No. The dogs also were a part of an interesting incident about 500 years ago, when the Spanish conquistadores invaded South America. There was a certain very dangerous Leishmania species here, Leishmania infantum, which did not exist in South America, but it was brought over with infected dogs from Portugal. We know today they came from Portugal. And since then, this Leishmania species causes a lot of problems and disease and death in Brazil, for example, and other parts of South America. So I entitled these slides Leishmaniasis in Ancient Times. So one could think, okay, it's not a big problem anymore. Unfortunately, it's just the opposite. I mean, you've noticed that we are in the middle of a climate change period where we have what we call tropical diseases that were never coming to Europe. Now they're coming to Europe, and we call them emerging diseases. And as a matter of fact, Leishmania and Leishmania infection, Leishmaniasis disease, is considered an emerging parasitosis in Europe. First cases of the very deadly bad visceral form have been seen in Greece. And of course, the climate change helps these insects to go towards the north. Then you have massive population displacement right now due to all these migrations. So it is clear that Europe will face eventually big problems with these parasites. I hope not as big as COVID, but fortunately, the European community invests a lot of money in Leishmania research because they know we need to act now and not when it's too late. And here's just some headlines from going between, Nepal to the United States, over Africa. And there's recurrent epidemics all the time going on of this disease. Even if you go to the Moroccan desert, be very careful. That's usually how Europeans come back with a very bad skin infection. What makes this even worse is there's absolutely no vaccine available. And after 25 years of research on vaccines, not me, but others, it's clear that it's not going to happen anytime soon. And the chemotherapy, so the drugs that we have against the parasite, they are failing because these parasites can very easily evolve and adapt to the drug and survive any kind of drug treatment. So it's a real problem. Okay, hang on. So, how was the parasite discovered? Well, first we had this Dr. William Leishman, a Scottish army doctor, who in Scotland received a lot of dead bodies from India, from, of course, was Commonwealth or the British Empire. And many soldiers died by this black fever or kala azar. And so this doctor was dissecting, and he already found that there was a correlation between the people dying and the liver and the spleen being swollen. And then another doctor, I think he's a British or English doctor, Mr. Donovan, he looked in the microscope and he discovered that inside your bone marrow, there's a lot of macrophage precursor cells. And always when a person was dying with black fever, inside these macrophage precursor cells in the bone marrow, he found that inside these cells, there are these little bodies. And they indeed were called for over 120 years Leishman-Donovan bodies, and they were diagnostic for the disease. Today, we know, of course, it is not just any kind of body, it is the intracellular form of the parasite inside the macrophage precursors, because it infects also macrophage precursors. So this is how the mammalian stage of the parasite was discovered. And then just to conclude the life cycle, we have these two brothers, Edouard and Etienne Sergent, who were working at the Pasteur Institute in Algeria some time ago, and they found that it's not bad water, because this is what people thought, oh, you drink bad water, you get Leishmania. They identified this little sandfly as the carrier of this form of the parasite. So the life cycle was, of course, identified, and this is now like a typical life cycle how we see it. So you have two different types of hosts. You have an insect host, which are these sandflies. You have vertebrate host, rodent, dog, human, cat, cow, whatever. In one part you have this motile flagellated kind of form that is more like needle shaped, and then once it's introduced, these parasites are taken up by macrophages, where they proliferate and of course cause the very bad immunopathology. So here's just again the team, and I will give you a very brief overview of the projects we're working on. All the projects in my lab are really working on studying the interaction between the Leishmania and its host cell, the macrophage. There's so many questions we don't understand. For example, how can this insect form sense that it's now in a macrophage and develop into this intracellular form, which not only the morphology, the structure is very different, the whole biology is very different. Another question is, how is it possible that the macrophage cannot work properly anymore and is literally paralyzed? And so this is on this side, the host, what we call subversion by the parasite. And finally, we have the middle team, which really links both. Here we ask... There is a microphone on, maybe? Okay, perfect. And so there is a third part of our program which investigates how the parasite interacts between being inside his vacuole with the host cell via the release of these little vesicles. This is very well established. All kinds of eukaryote cells release vesicles into the environment. And so we believe that Leishmania releases these vesicles in order to change the macrophage to become like a very nice host cell for these parasites. So the first part of the scientific talk will be, of course, the movie title, Dressed to Kill, where I would like just briefly show you a few experiments that I did, maybe, hang on, 25 years ago. So it seems old, but it's still a pretty cool story, you know. And it's literally an example how to identify and validate what we call a virulence factor. I will show you. So the virulence factor that we are looking right now is called LPG. It's just like a short form. I will tell you what it is. And it can be visualized on this experiment. We just illuminated with fluorescence this LPG structure, which covers the entire parasite surface. And I showed you already before, it creates this kind of sugar coat. It's sugar molecules that are anchored with a lipid anchor in the membrane. And so this, of course, this surface is the interface between anything that happens between the environment of the macrophage and the internal environment of the parasite. So what is the sugar coat? Here's just a schematic overview. This is the lipid bilayer of the parasite membrane. So we're talking about the parasite now. And the parasite is covered by this lipid anchored sugar molecule, which is a very long and very complex sugar molecule. And because there are so many molecules on the surface, they literally create this kind of shield against all kinds of macrophage attacks. And so first question that one can ask, okay, this is a very abundant, very smartly localized molecule. What's the role of this molecule in allowing these parasites to survive in the macrophage? And of course, this means the question, is this molecule LPG a virulence factor? Now virulence factors are defined by microbial characteristics that can enhance the ability to cause disease. Anything that allows adhesion of a pathogen to a tissue or allows it to invade a cell or resist against an immune response and so on can be considered a virulence factor. But how can we make sure that we really can define this as a virulence factor? And there we have what we call the Koch's postulate. Now, Koch is a very famous German microbiologist, Robert Koch. He lived more or less at the time of Louis Pasteur. I think they were pretty much competitors, just like sometimes it's still today. And so Robert Koch, he said, okay, this was about at least 100 years ago. He said, in order to really understand how a molecule helps the microbe to make us sick, here's what we have to do. First of all, we have to identify the factor that we think is involved in virulence. Then, second, we need to remove the factor. And if the microbe then loses infectivity, that's already a very good hint that this factor is important for the microbe to infect us and make us sick. But then the most important step is that you take the microbe without the factor and you have to add the factor back, and the microbe with the factor added back should be, again, disease causing. So this is really find it, remove it, and add it back. And if this correlates with infectivity, you have validated that it's a virulence factor. So I was interested in investigating whether this long molecule is a virulence factor. And so I used a genetic approach based on homologous recombination, which is just shown up here. I'm not going to go into details, just to tell you that what we did, there is a certain gene, which we call LPG1, which is very important for the biosynthesis of the glycolipid. Now, I used homologous recombination to delete this gene from the genome of the parasite. And lo and behold, these parasites were literally naked, as shown here by the absence of green, and the green color being, of course, the molecule LPG. And then once we removed this LPG from the parasite by destroying an important gene, ultimately, I used this parasite and I added back the very gene that I removed on a little construct shown here. And lo and behold, these deleted parasites now, with the additional new gene, they re-express the LPG on the surface. And so the first thing that I did was, of course, to ask the question, how are these parasites doing in macrophage and mouse infection? And so here is a typical mouse infection experiment, a very simple one. You take these poor little creatures, and all you do, you take a wild type, what we call a knockout, and what we call an add back. You take these parasites and you inoculate these parasites in individual mice under the skin of the food pad here in the back. And what happens is, just like in the human, this is a cutaneous Leishmania species, parasite species, just like in the human, the injection of the parasite under the skin will produce a lesion. The more and the faster the lesion, the more virulent and disease-causing these parasites are. And so here you can see the number of days after I injected the parasite in the food pad. And on the y-axis, you have the size of the cutaneous swelling, what we call a lesion, as a function of the infection time. And so, of course, injecting the wild-type parasites, you see that very fast, after 2 weeks or so, we can see that the skin is swelling and the skin is full of parasites. However, this parasite here, the LPG1 knockout that doesn't have the LPG, look what happens. It is extremely what we call attenuated. It is very weak. And only after 60 days, there is a small little response which never goes up to the same level. So this was already a very good hint. According to Koch, we identified already a factor that is maybe important. Second, by removing the factor, we showed, yes, it is correlated to infectivity. And of course, now the third postulate, to add it back, and this is the experiment where I added it back. In red, again, you see the attenuated parasite, just like here. And in purple, you see now these parasites where the LPG was re-expressed and they regain in virulence. They're not completely as strong as the wild type, but they're much stronger as these knockout parasites. And this together establishes and fulfills the Koch's criteria to say LPG now is really defined as a very important virulence factor. The same can be done not only in mice, but you can also just take purified macrophages from the mouse, and this is shown here. So here you see a macrophage in a microscope image that is invaded by these parasites here. And after a few days, you just look these intracellular parasites, how they grow and develop. And again, we've seen that the wild-type parasite causes a persistent infection of the macrophage, whereas the knockout parasite is just destroyed. So LPG is a true virulence factor. And the next question we ask is, yeah, okay, but what is LPG doing to help these parasites survive? Now, you remember this picture from the funny introduction. So these parasites, once they're inside a macrophage, again, they have to survive many different steps. First step is already before they go inside the macrophage. There's outside in the bloodstream. And so in our blood, we have many toxic components. And one toxic component is called the complement system, which is nothing but a protein cascade that can introduce a tunnel in the membrane of a microbe. It's a really fascinating system. They recognize the microbial surface, go there, and there will be a hole formed, and the microbe just leaks out. So complement, the parasite needs to survive complement. Then it needs to survive this oxidants, toxic oxidants produced during the uptake. It needs to survive the lysosomal fusion, and then eventually, block the immune system later on. So what I did, I took my parasites, the wild type, the knockout, and the AdVac, and I was designing experiments to test step for step whether this LPG is important for one or the other resistance. So the first thing that we did was looking into the complement system. It's a very simple experiment. I added a rat dye into the culture medium, and this dye was not taken up by the parasite. But if the parasite has holes, the dye will go in. And so what I did, I had these parasites in this medium, that is with this red fluorescent molecule. I added human serum, which was mine because there was no other way. So I went to the university doctor. This was 25 years ago. Today, you cannot do this anymore. So I took my own blood, I took the serum, and I added this to the culture. If the parasite is resistant, it will not turn red. If the parasite has holes and is sensitive to the complement, it will just be full of red. And we can monitor and quantify this using a very sophisticated fluorescent machine called FACS. I'm not going to explain it, but I will show you the result. So a FACS analysis can show you the level of fluorescence on the x-axis and the number of cells on the y-axis. So if you see this kind of mountain, it means the surface under the mountain corresponds to all the cells that are analyzed for their fluorescence intensity, which will be plotted here. It's complicated, but you will see it's a very simple thing. So here we have the wild type parasite that has the sugar coat. And so in black is a control, no serum added, but in the presence of the red fluorescent dye, and you see, well, they're not really very red. Then I added the serum. There's a little tiny thing. You see that there's a little shift, a little bit more red, but nothing really dramatic. And now what happens if we now do the experiment with the LPG1 knockout? Again, without any serum, they're just perfectly fine. They are happy and alive in the culture medium, even with the red stuff around it. But what I did now is I added my serum, and every 2 minutes, I was measuring the level of red. And this is a little animation, 25 years old. And you will see how the population of the parasite, little by little, there is more and more red coming up. So what you see here is that the population of parasites we analyzed here goes from not being red, all the population turns to be red. And this was the end of the experiment. And you can, I think, see the notable difference. And so this showed that LPG is absolutely crucial for complement resistance. In the wild type, nothing happens because there's the sugar coat. And so these tunnels, they cannot really reach down to the plasma membrane. Whereas in the naked parasite, the tunnels reach the plasma membrane and poke holes that kills the parasite. So that was the first. Then we go on to the next, which is these oxidants that are produced when the macrophage takes up the parasite, which normally should be very toxic. And so here's a really fancy experiment. Well, I will show you, I explain it. So in thick is, of course, the wild type parasite with the big coat. And then this is a macrophage. And here it's a thick macrophage because it's a wild type macrophage as well. You will see in a second why. So you can infect a wild type, you can use a wild type parasite to infect a wild type macrophage. You can, of course, use a knockout parasite and infect a wild type macrophage. I've showed you this experiment just a few slides ago. These LPG knockout parasites, they're not doing well. They're killed in these wild type macrophages. But now comes the real experiment that will allow us to test how important are oxidants in parasite killing and in the function of LPG. So what we did, we used the mutant parasites and we infected a mutant macrophage that we got out of a mutant mouse. And this macrophage cannot produce the oxidants anymore during the uptake of any kind of particle parasite or whatever, you know. And so the question was, what will happen? We know that the parasite without LPG is killed in a normal macrophage. Can the parasite survive in the macrophage without oxidants? And so the result is shown here. So here you see the survival. In the upper panel, I show here, these are wild type macrophages that were infected with wild type in black parasite or with the LPG knockout parasites. And as we have seen before, the wild type parasite in a normal macrophage is doing rather well. The knockout parasite in a normal macrophage is not doing well at all. However, if you do the same experiment with these mutant macrophages that are shown here, the wild type is doing more or less the same, but look what happens with the knockout. The knockout is totally restored for its capacity to survive inside the macrophage. So this clearly shows that these parasites in the wild type macrophage are vulnerable to oxidants. And so LPG, that's the only thing that they miss, is important for oxidant resistance as well. So I'm going to skip here. And so, Anna, you keep the time. I don't have any idea how much time it is.
Oh, I think it's okay. I think you can go on. Yeah.
Yeah. How long?
Maybe like 5 to 10 minutes.
5 to 10 minutes. Yeah, okay. I will make it very fast. So a first conclusion, and this was the longer part, of course, the rest will be fast. What we showed is that Leishmania is really tough. And the secret of Leishmania toughness is its sugar coat. And so this sugar coat allows the parasite to already resist against the blood complement and also to resist against these very toxic oxidants, which then allows the parasite to survive in the macrophage. And so once the parasite is in the macrophage, I told you this form that is more the insect form, this long shaped with a flagellum, it turns into this form that is more oval shaped that we call amastigote. And so this is the form that really grows inside the macrophages. And so, of course, we were very interested, and this is what I do today. As I told you in the introduction, we're really looking into what is the interaction between these parasites and their host macrophage. And so how do we do that? You know, this is not a trivial undertaking. So we use mice, unfortunately. I mean, we all love animals, me too, but in order to find something to cure humans, unfortunately, it's the only way we can go. And so what we do here is we establish a infection system where you can see here in this, it looks like an egg, you know, in a pan. This is the cytoplasm of the macrophage. I think if you see my mouse, maybe it's okay. This kind of oval-shaped thing is the nucleus of the macrophage. And this gigantic blob here is the vacuole. So this, space that hosts the Leishmania. And what you see as these little pebbles on the membrane of this vacuole are indeed the Leishmania. And so what we do in order to establish the system, we extract bone marrow from one mouse, the white mouse, that allows us to establish the macrophage culture. And in parallel, we have infected these mice where the parasite grows very well. And so we can obtain lots of parasites right from the infected animal. And then we incubate the macrophages from one mouse with the parasites we obtain from the other mouse. And we can also visualize what happens in a very fancy way. So here, for example, our parasites, so these are these little berries here, they are red, fluorescent red, because we have overexpressed a transgenic marker that turns these parasites permanently red, so we can see them in red under the microscope. We have this huge green ball, which is the vacuole. This is a chemical that is turning green every time that there is an acidic pH. And of course, this phagolysis or these vacuoles are extremely acidic because it's like a stomach. And so this is what we use to visualize the house of Leishmania. And then in blue is another staining that allows us to visualize the macrophage nucleus. And so one very important study that we recently did is shown here. You will see, I really don't go into details. It's just to give you a little bit more of an edge saying like, oh gosh, this is too complicated. It's good that you know it's complicated. So there is, the macrophage is not really without defense. The macrophage is full of defense systems and full of arms, you know. And also, of course, it can sense the infection and tells the immune system, go wild, you're infected. And so one system that is really important for the macrophage to signal to the entire immune system, danger, there is an infection, is a protein complex inside the macrophage that is formed in response to products of microbes. And this complex this is like 4 proteins that are forming this complex. It's really like a machine. It really looks like that. There is some electron microscopy. It's beautiful. It really looks like this machine. And this machine is called the inflammasome because it induces inflammation. It allows the production of a hormone, I want to call it, a cytokine, that is turning on the immune system to be antimicrobial and inflammatory. It's called IL-1B. And so the big question we had was here, just a very simple question. What happens? You have on one side this super important complex inflammasome in the macrophage, but then you have also Leishmania in the macrophage at one moment. What is Leishmania doing to this inflammasome? Is Leishmania activating the inflammasome, as it should happen because it's a bad parasite? Or on the contrary, is Leishmania suppressing this inflammasome? And I'm not showing you data on this. I just tell you that Leishmania goes in totally silent. And even worse, even if you want to activate the inflammasome with some bacterial products, which are very well established, Leishmania totally blocks, inactivates the inflammasome, and thus the very important inflammatory response that normally would kill Leishmania. So I'm not going to show you any data. This has been recently published. It was a big story. But what I want to show you is that this inflammasome is not only helping the immune system to be activated, but eventually the activity of this inflammasome causes the death of the macrophage that has the inflammasome activated. And again, I'm not going to go into details, but I want to show you some fancy pictures and movies. What happens is that there is an inflammasome-dependent form of cell death, which we call pyroptosis. And in this process, there is this molecule generated that inserts in the membrane and forms a hole in the membrane. And so this is a hole formed by the macrophage to himself, which causes leakage. And it seems to be a very important, again, potent immune stimulant because the cytokine can be released. And this molecule even can insert into the microbial membrane and also eliminate microbes as well, similar to the complement system. And so, of course, we said, what the hell is going on with Leishmania and this during pyroptosis? Is there pyroptosis? And if yes, is the Leishmania sensitive to this pyroptosis pore-forming molecule? And so this is a little experiment. I think I want to show you first the movie, and then we're going to look very briefly, and then I'm nearly done. So this is a macrophage that you see the blue nucleus. In green, 1, 2, 3, there are 3 green bubbles, which are these vacuoles that contain the parasites. And of course, you can see here in reddish yellowish, the parasite itself. And so when we looked in the microscope, what happens in these macrophages when we induce pyroptosis by adding some of microbial products, and look what happens. So immediately the green goes away, and I don't know if you can see this, the parasites are literally ejected like some escape pods of Star Trek movies. I will show you this again. The green goes away, parasite ruffle up and bloop, they're just exposed to the outside. And so as a matter of fact, what we see, this is basically a time course experiment where we, at time 0, we activate pyroptosis, or we try to, and it's working. It can be activated. And very fast, the green level goes down, which means this vacuole starts to deteriorate. And in parallel, what we measure in the black curve is parasite death. And so, yes, more and more parasites are dying when they start to undergo pyroptosis. It's normal. But what we were really intrigued was this exposure of these parasites. And so we did some fancy electron microscopy that is shown here. So here is the same system I just showed you in the movie. And we processed it here for what we call a transmission electron microscopy. So you get a 3D kind of form of what happens. And the parasites are expelled, but they're all anchored, which is shown here on this EM staining. They're all anchored in the membrane, and they all show the same position. They have this little flagellum, looks like a nose pointing outside. And of course, we were very intrigued by this finding. It seemed that the parasites were not affected by the macrophage lysis at all. And as a matter of fact, we could show this in follow-up experiments. I'm not going to go into detail. I just tell you that the Leishmania is totally happily coming out from these dying macrophages. And we even believe that this kind of cell death may even help the parasite to disseminate and go into new macrophages. So I think this is the end. Just a take-home message. I hope you know what Leishmania is, you know what it does. It's very resistant, so very tough, but it's also very smart because it knows exactly how to subvert our immune system. This is why I say to some of my students when I give lectures that Leishmania is the best immunologist. And so studying Leishmania helps us even to understand our own immune system. The team again, my funding, and I thank you for your kind attention.
Thank you so much. Actually, just before you finish, there's a question from Mr. Damon. If you received a blank check, any amount of money to do more research, how would you spend it? What would you
build a lab in Numea and move. No, I'm just kidding. I'm just kidding. Yeah, this is a typical question, of course, that we often get when we have interviews at the highest level of the funding agencies. I think the money should be spent where the disease is, and this is unfortunately not always the case. So what I told you right now, this is representing, I don't know, 5% of what we're doing in the lab. There is a big project that I'm animating with all the Pasteur institutes that are localized in the endemic area of this disease. And so I'm running a big European consortium with a lot of money in order to work with these people, make them work together between themselves as well. And so for me, the biggest, I would say, the biggest catastrophe that happened in science is that people tend to work on animal systems. We have the mouse as a model system, you know, and we're lucky because Leishmania really infects the mouse, but still, the mouse is a very lousy system. It's so different from the human system in terms of immune response that many vaccines and many drugs that are working in the mouse are never working in the human. And this is the biggest catastrophe, that somehow science has become a little bit too convenient. Let's take the mouse. We can go to the pharma company and buy all the antibodies against the mouse, and work to do. So I think the money should be spent, not necessarily in my team, but there should be money spent to really allow the scientists to work with human samples, to work with the patient, and to really work with the real disease and the real parasites. Because what we have, not anymore, but many people that are working on microbes, they're just having this microbe that they keep in culture since 20 years. That's not a pathogen anymore. Because they evolve in culture very much. And so if I would have to spend money, maybe not for my lab, but for science in general, I really would spend it on re-inciting human clinical research and going in the field, working with the scientists in the field so that they have access to our fancy machines and we can instruct them how to use them and collaborate on this side. That's what I would say.
Just to finish off, I think there's just two very last questions. Alma is asking, are there any cures available if you do get Leishmania?
Yes, there are cures available. One cure is a funny cure because it kills the person that should be cured. It's a very toxic treatment invented about 120 years ago. It is based on arsenic. And now I think you all know that arsenic is really toxic. And so it's just a little bit more toxic to the parasite and to the human. There are other more modern treatments, but as I said, the problem really is we are running out of time and it's like an arms race. We invent a new drug and the parasite invents a new resistance mechanism against the drug and the drug is useless. And so we really always try to be one step ahead. This is why a lot of money is invested today, where we still have a few treatments that are working, but today is a lot of money invested to find new treatments. And this is a big part of what we do in the lab is to find new treatments.
Thank you. And one very last question from Tessa is, would Leishmania be able to cause a pandemic as big as the COVID-19 pandemic?
Whoa, I wish not, just kidding. No, you know, COVID, of course, you have to imagine is spread by droplets, you know, so already there you have the answer. Leishmania needs to be infecting a sandfly. The sandfly needs to live in a certain environment. It cannot live in Paris, the sandfly, that's for sure. Then it needs to bite a human, which maybe the sandfly rather likes to feed on dogs because sandflies have feeding preferences. You know, maybe the human is not such a nice, you know. So the answer is clearly absolutely not. But given the fact that Leishmania infects 12 million people, every year there are 2 more million people infected. Hundreds of thousands are dying every year, and you guys never heard about the disease. So we are, I think that the global health impact of Leishmania is huge. Of course, right now, COVID is going crazy. Everybody shuts down the country. So the impact on economy is even bigger than Leishmania. But if you would calculate over the past 50 years, how much economic damage Leishmania was doing because people die, people get sick, you know, it's much bigger than COVID today. But a pandemic, I don't think so.
Well, thank you very much. Thank you so much to everyone who came, and thank you so much, Dr. Spaeth. It was honestly such a blast.
Again, thanks for the questions and for listening, and remember Leishmania. Good night.
Well, with that, thank you so much. I hope everyone has a nice night. Thank you.
Yes, you too. Bye. Bye-bye. Thank you
very much.
Thank you. Thank you so much.
We learned a lot.